Welcome to Let's Learn Plants. I'm so thrilled you're joining me for this explainer today. We're going to dive into a seriously fascinating aspect of plant physiology. We're looking at how plants perfectly, and I mean perfectly, synchronize their carbohydrate production with the exact amount of light they receive. Specifically, we'll be exploring the incredibly intricate regulation of carbon fixation reactions in photosynthesis.
It's a really simple question, right? Why don't plants make sugar in the dark? But it hits right at the heart of our tutorial today. You see, the carbon fixation reactions, which we know as the Calvin-Benson cycle, require massive amounts of cellular energy in the form of ATP and NADPH. If a plant just kept running this cycle after the sun went down, it would drain its hard-earned energy reserves completely empty. There'd be nothing left. So, to survive, plants have evolved a series of ingenious master light switches. These mechanisms make absolutely sure that the carbon fixation machinery is strictly turned on during the day and decisively shut off at night.
Okay, let's dive into this. Here is our roadmap for the explainer today. We're starting with the carbon fixation challenge. Then we'll explore 4 distinct regulatory mechanisms: regulating Rubisco, the Ferredoxin-Thioredoxin System, chloroplast ion movements, and CP12 complexes. Finally, we'll wrap the whole thing up with regulation in C4 plants.
Section 1. The Carbon Fixation Challenge. Warming up the machinery.
Picture the exact moment the sun rises and the very first rays of light hit a plant leaf. The plant is suddenly just bathed in energy, but its biochemical machinery inside the chloroplasts isn't instantly running at top speed. It's kind of like a cold car engine on a freezing winter morning. It needs a minute to warm up. We call this warm-up phase the induction period. It's the initial lag right after illumination where CO2 fixation gradually increases. Now, why the lag? Well, 2 things actually need to happen. First, the light-dependent photosynthetic enzymes physically need time to activate. And second, the concentration of the Calvin-Benson cycle intermediates has to build up. During this induction period, the cycle uses all 6 of the triose phosphates it produces just to regenerate RUBP. It's only after reaching a steady state that the cycle shifts gears. Then, 5 triose phosphates are used to regenerate RUBP, and that 6th one is finally siphoned off to actually make starch in the chloroplast or sucrose in the cytosol.
Now, plants do have long-term regulation, which involves turning specific genes on and off to change the total amount of enzymes. But our focus today is on short-term regulation. This is all about incredibly rapid post-translational modifications. Light-dependent mechanisms quickly target and alter the specific activity of 5 key enzymes. Let's carefully note them. We've got Rubisco, fructose 1,6-bisphosphatase, pseudoheptulose 1,7-bisphosphatase, phosphoribokinase, which we'll just call PRK, and NADP-glyceraldehyde 3-phosphate dehydrogenase, which we'll refer to as KDH.
Section 2. Regulating Rubisco activity. Fixing the jammed machine.
Now, what's really interesting about this process is how our heavy lifter of carbon fixation, Rubisco, essentially jams its own gears. For Rubisco to even be active, it absolutely needs an activator CO2 molecule to bind to it, a process we call carbamylation. But what happens if it loses that activating CO2? Well, it becomes uncarbamylated, and in this state, its substrate, RUBP, can bind to the active site incredibly tightly. This creates a dead-end complex. The enzyme undergoes a conformational change and is completely inactive. It's literally jammed.
So how in the world does the plant fix this jammed machine? It actually brings in a mechanic, an enzyme called Rubisco-Activase. This process requires ATP and happens in a very specific step-by-step sequence. First, ATP hydrolysis primes the activase. Second, this primed activase physically interacts with the jammed Rubisco. Third, it forces a conformational change in Rubisco, lowering its affinity for that stubborn RUBP. Fourth, the RUBP finally dissociates and falls away. And fifth, reactivation. Now that the active site is totally clear, a CO2 molecule and a magnesium ion can jump in, bind, carbamylate Rubisco, and put it right back to work fixing carbon.
Section 3. The Ferredoxin-Thioredoxin System. The electron relay.
Let's move to photobiobroxionation and see how this builds upon what we just learned. If Rubisco-Activase was our mechanic, this next system is basically a direct electrical wire connecting sunlight straight to the enzymes. It's a continuous electron relay. Light strikes the plant, driving the transfer of electrons from water to a protein called ferredoxin, reducing it. This reduced ferredoxin then passes its electrons down the line to an enzyme called Ferredoxin-Thioredoxin reductase. This reductase then hands the electrons off to a small protein called thioredoxin. Finally, this reduced thioredoxin physically interacts with our target enzymes, reducing their regulatory disulfide bonds. It essentially snips those molecular locks right off, popping the enzymes into their active shapes.
And which enzymes get activated by this thioredoxin relay? It's 4 of the 5 key enzymes we listed earlier: fructose 1,6-bisphosphatase, cetoheptulose 1,7-bisphosphatase, PRK, and GAPDH. The real beauty of this system is its immediate reversibility. The exact moment darkness falls, electron flow from the light reactions stops cold. Thioredoxin becomes oxidized, those disulfide bonds in the enzymes immediately reform, and carbon fixation shuts down completely. No light, no wasted energy.
Section 4. Ion movements in chloroplasts. Shifting the balance.
And this brilliantly illustrates how our third regulatory mechanism relies on the actual physical and chemical environment of the chloroplast itself. Notice how, when illuminated, the light reactions pump protons from the stroma straight into the thylakoid lumen. To balance out this electrical charge, magnesium ions flow out of the thylakoid and into the stroma. This shift creates a highly specific environment in the stroma. You get a high pH, meaning it's alkaline, and a high magnesium concentration. And guess what? This is the exact environment required to activate Rubisco and our other key enzymes. When the lights go out, the whole flow reverses. The stroma's pH drops, the magnesium leaves, and the enzymes rapidly deactivate. It's just so elegant. Section 5, CP12 and Supramolecular Complexes. The dark shutdown.
All right, so we've talked about chemical modifications and environmental shifts. Now let's look at spatial and structural regulation. This involves a fascinating little protein called CP12. Remember, the plant's ultimate goal in the dark is to prevent the wasteful consumption of ATP and NADPH. CP12 achieves this by acting as a physical clamp. In the dark, CP12 is oxidized, and in this state, it literally wrangles 2 major enzymes, a specific form of GAPDH and PRK, and binds them together. This creates a massive, inactive ternary supramolecular complex. It is literally like taking 2 heavy factory machines and locking them away in a storage crate for the night.
The absolutely crucial point here is understanding how this CP12 clamp affects the different versions of the GAPDH enzyme. For instance, the A4 homotetramer isoform is the one we just discussed. In the dark, it gets locked tightly into that inactive ternary complex. But the A2B2 heterotetramer isoform acts differently. Its regulation is actually autonomous. In the dark, it forms this larger inactive A8B8 oligomer, which is regulated by a disulfide bond on its B subunit. But what happens when the sun comes up? The Ferredoxin-Thioredoxin system kicks into high gear. Reduced thioredoxin smashes these complexes apart. It cleaves the disulfide bonds in both CP12 and the A8B8 oligomer, freeing those active A4 and A2B2 forms to begin fixing carbon for the day.
Section 6. Regulation in C4 plants. Adapting the rules.
So far, we focused pretty heavily on standard C3 photosynthesis. But C4 plants, like corn and sugarcane, well, they've evolved specialized anatomy to hyper-concentrate CO2. So naturally, they've had to adapt these light switch rules to match their unique setup. They manage their enzymes depending on the exact time of day using complex thiol disulfide exchanges and phosphorylation. For example, NADP malate dehydrogenase relies on that familiar Ferredoxin-Thioredoxin system we covered earlier, so it's activated by light. But the second enzyme, PEP carboxylase, is regulated by diurnal phosphorylation. A specific kinase phosphorylates it, which actually makes it active at night, protecting it from being inhibited by malate. Finally, pyruvate phosphate dikinase, or PPDK, relies on a bifunctional enzyme that deactivates it in the dark through phosphorylation and restores its activity in the light by removing that phosphate group. It is honestly a beautifully choreographed metabolic dance.
Which brings us to the end of our explainer with a final, really provocative thought. We've seen how meticulously plants manage their energy, turning enzymes on with light and shutting them off in the dark to save those precious resources. If we can map out and fully understand every single one of these master light switches, could we use agricultural biotechnology to re-engineer them? Could we tweak the induction period or Rubisco-Activase to make crops wake up faster and grow more efficiently to feed our growing world? It's a thrilling question to leave you with. Thank you so much for joining me today to learn plants and keep exploring.